Phospholipids form an essential part of the cell membrane, forming the bilayer structure. They have a similar structure to triglycerides, but the third hydroxyl group on the glycerol molecule is bonded to a polar phosphate rather than a fatty acid. We describe these molecules as amphipathic, where the polar phosphate group is strongly attracted to water, which we call hydrophilic. The fatty acid chains are non-polar and therefore repel water, which we call hydrophobic. This means that when phospholipids are placed in water, they can orient themselves by either forming a monolayer where the hydrophilic heads are facing into the water and the hydrophobic tails away from water, or a bilayer where the hydrophobic tails again are facing away from the water. There's another way in which phospholipids interact with water, which we can see in the micellar structure. A micellar consists of a single layer of fatty acids arranged in a globular fashion. The polar heads point towards the outside and interact with the aqueous environment, while the hydrophobic tails are packed into the inner space and are prevented from interacting with the aqueous environment. The composition of phospholipids contributes to the fluidity of the cell membrane. If they are mainly saturated fatty acid tails, then the membrane will be less fluid. If they are mainly unsaturated fatty acid tails, then the membrane will be more fluid. If the membrane is too fluid, it will let more molecules through it, and if it is not fluid, it will prevent molecules from passing through it. Phospholipids control membrane protein orientation. Weak hydrophobic interactions between the phospholipids and the membrane proteins hold the proteins within the membrane, but still allow them to move within the layer. Aside from phospholipids and triglyceride, other lipids include steroids, cholesterol, and waxes. Cholesterol is vital in the manufacture of vitamin D and has a role in the cell membrane. Just like phospholipids, cholesterol is an amphipathic molecule having both hydrophilic and hydrophobic regions. Cholesterol's hydroxyl group is hydrophilic and aligns towards the phosphate heads of the phospholipids. The steroid ring and the hydrocarbon tail is hydrophobic and associates with the phospholipid tails. Cholesterol affects the fluidity and permeability of the cell membrane. As we mentioned before, if the membrane is too fluid, it will let more molecules through it, and if it is not fluid, it will prevent molecules from passing through. Cholesterol disrupts the close packing of the phospholipids, increasing the flexibility of the membrane, preventing it from stiffening or crystallization. As the concentration of cholesterol increases, so does the fluidity. It also does something very interesting at different temperatures. At high temperatures, cholesterol helps reduce fluidity, and at low temperatures, it increases the fluidity. In this way, it has an important role protecting membrane fluidity from fluctuations in temperature. It also acts as a barrier, fitting in the spaces between phospholipids, preventing water-soluble substances from diffusing across the membrane. Steroids contain a ring structure. In animals, many hormones are steroids, including testosterone and estrogen. These are also made from cholesterol. Waxes have a similar structure to triglycerides but have fatty acids bonded to long alcohols rather than glycerol. At room temperature, they are usually solids and provide protection and waterproofing on the surface of leaves and insects. Hi, my name is Mr. Science, aka Salim. If you're new to the channel, please remember to like and subscribe, and for more teaching resources, you can visit my website at www.mrscience.co.uk.